going to cover genetic resources today. I've talked a lot about a lot of different shapes and colors that we see in horticultural crops. But really, if we want to make some genetic improvements or some genetic changes to crops, we need to know where to go to get the resources that we can use in these breeding programs. I've spoken an amount about some consumer aesthetics, such as color, but if you're able to go and look at genetic resources, you can see some of the huge variation that may actually be out there but not present, for instance, in supermarkets or a lot of commercial varieties. So to look at raspberry, for instance, you could actually select genotypes that go anywhere from the yellow raspberry and have all of the color variations through to the black raspberry if you're looking to vary the color on this type of crop species. But when we look at commercial crop plants, they are very different from a lot of the genetic materials that may be out there, or a lot of the naturally evolved crop types. Domestication has largely been based on the selection of rare mutants over time, and mutant genes that wouldn't be very competitive in a natural environment. For instance, selection of large fruit type or large seed type that may be very good for agricultural or horticultural purposes, but in a natural environment, the wilder type of plants would compete against them more favorably, and they wouldn't perform particularly well. So a lot of these improvements are great for agriculture, but not advantageous for survival in the wild at all. So oftentimes some quite simple genetic changes can make some profound changes to the crop type for harvesting for food. But oftentimes there's a lot of changes that have occurred over time in those crops that we sometimes take for granted and we don't always think about when we're just thinking about issues like disease resistance or yield or maturity. So I put this table up from GEPT, which just compares some traits in a wax bean Midas. A wax bean is a yellow, fresh harvested snap bean. Just to show that if we were to take a wild common bean that was collected, some of the differences that we might see. So for instance, the fiber in the pod suture and wall, you get a lot of fiber present in the wild type, but over generations this has been selected against in the snap bean type because we like the beans to snap and basically have no fiber in them. We also look at the seed quality. In the wild type, we're only getting 70% germination of the seeds. If you try and sell a seed commercially with 70% germination, you're going to have a lot of problems with commercial growers. Basically, seed has to have pretty much 100% germination, which it does in most commercial types. So increasing for high germination is a factor that might be selected for in a domesticated variety over a wild. We also have determinacy. An indeterminate plant is one that has a vine type of growth habit, keeps growing quite tall, where a determinate plant is a type of a self-pruning sort of plant, so it has more of a bush type of habit. The benefit of having a determinate habit is that most of the fruit, or in this case the bean pods, will mature at the same time. Where the vine type, they may develop pods at different stages over time. So when we develop determinate bean types like this, it opens it up for machine harvesting which drastically reduces the cost of bean production. This is also the case, for example, with processing tomatoes that are machine harvested. Twine-in, we have the wild beans will twine up the stake. We'll have a lot more nodes, which means we have a lot more trifoliate leaves that come off the stem. Where here we have a much smaller bush type of plant with a smaller number of nodes. We have a smaller number of pods on the bean type at 14 compared to 43, but these pods are much bigger, with a weight of about 19.5 grams compared to 3.5. And also the size differences also vary a lot. And then the maturity has been brought up considerably, days to flowering and days to maturity. And we have an absence then of things like seed pigmentation. Consumers want the white seeds in snap beans. And they have a much higher harvest index of about 62%. So about 62% of the weight of the plant is actually harvested for consumption. So this just gives an example of if we were to go to a wilder type of germplasm, a lot of the traits that we may find in it that are not present in commercial cultivars, and then the difficulties that we may have removing these deleterious traits in a backcrossing program. It's not just the linkage to the perhaps disease resistance gene we may be working with, but as we backcross to a commercial type, we are kind of building on the back of a pyramid of a commercial variety that has been produced over a long period of time. But any number of these genes could have a lot of deleterious linkages associated with them. So during a backcrossing program, we don't just deal with the negative linkages of our trait of interest, but a whole host of other traits depending on the crop type. So it's something that needs to be taken into account when working with wilder germplasm, because it means that the breeding time to actually introgress or move a gene from a wild type can take a considerable period of time. The MI gene in tomato, which controls root knot nematode, was known since about the 1940s. With breeding programs, it was worked on as a single gene, but it took almost 40 years to get rid of deleterious linkages before it really started appearing in commercial varieties. So this idea that we can go out there and find a gene of interest and then just move it straight into a commercial variety quite easily is maybe a little bit naive if you don't really understand a lot of the other traits of importance that may be present and selected for at the same time. But when we're going to look for diverse materials that we can work with, we oftentimes go back to areas where you basically have the origin of that crop species. The centers of origin that were proposed by Vavilov, a Russian scientist in the earlier part of last century, he basically hypothesized that where you have the most diversity of a crop plant was where the origin of that crop plant was based. And he did a considerable amount of work in the earlier part of last century, and the Vavilov Institute is still one of the largest germplasm repositories in the world. It is suffering from critical lack of funding at this point in time, which means that a lot of materials they hold are really at risk of being lost forever. And with so many commercial varieties being grown these days, it's very difficult to go out and collect the diversity that we could have done 50 or 100 years ago. But Vavilov proposed that there were 8 centers of origin, and you might have come across this theory in some basic biology classes. But the 8 centers were basically around the Mexican, Mesoamerican region, the Andean region, the Abyssinian or Ethiopian, Mediterranean, Near East, Central Asiatic, Chinese, and then the Indo-Malaysian type of regions. And you can see where some of the origins of crops are based around the globe from this. For instance, in the Andean region, we have crops such as potato and peanut as their origin. And in the Middle American region, we have crops such as corn and chili peppers. So these were new crops that were added to the world's, or at least the old world food supply around the 1500s. To split this down into the vegetables, fruits, and agronomic crops, we can see where the origin of a number of vegetables are based, where we have crops like carrot, turnip, and cabbage being based, having their centers of origin around the European region. Where in Latin America we're picking up crops like pepper, tomato, potato. We also have secondary centers where crops may have been moved and domesticated in a different region. So if you look at North America, a couple of the crops that have been developed here are Jerusalem artichoke and sunflower, for example. Also cranberry, I believe. This is an example of some of the diversity of the origins of fruits. You can see that down in the Middle Eastern region, we've got the development of crops like olive and plum. But as on a stone fruit basis, we're moving across to an Indian origin for cherry, and then going up to a Chinese origin for peach and apricot. So the development of the stone fruits aren't necessarily from the same place. They might have been domesticated in their origin with different populations throughout all continents, essentially. Some of the crops appear in more than one continent, so they have different bases. So, for instance, grapes originate both in the Mediterranean region, which is Vitis vinifera, but grapes also originate, for example, in North America, which are Vitis labrusca, which is a different species. This is also true of cotton and strawberry and some other crops, where there's actually different, basically, species of those crop types. You can see sunflower originating in North America. So it's useful to know this information, because if we want to go and collect a lot of diversity to use for genetic resources, then obviously going back to the center of origin is where we're going to find a lot of this diversity. So depending on where the crop type originates from, or where it has most of its diversity, this is where we're going to get a lot of variable germplasm that we can actually work with and use. More recently, Hawkes proposed the nuclear centers and regions, where he proposed there were basically 4 centers of origin, or nuclear centers, in northern China, the Near East, southern Mexico, and Peru. And the secondary centers were just regions of diversity, because he could not find information based on archaeological remains and wild progenitors that suggested that the crops actually originated in all 8 centers proposed by Vavilov. So he basically proposed 4 main nuclear centers, and then you have regions of diversity in microcenters where maybe a particular crop will originate in a particular area. So what are we talking about when we're talking about genetic resources? Oftentimes it's referred to as germplasm. Germ is Latin for life, basically, and plasm is a mold, so germplasm basically means life mold. So germplasm can basically represent any kind of living tissue that can be basically generated into a crop plant. So it isn't necessarily restricted to just seed, but it can involve such things as cuttings in the case of crop plants. They would all be considered germplasm because they can basically be regenerated into the original plant type. The germplasm that we would consider collecting or using is oftentimes based on materials such as landraces. These are crops that are selected within certain communities over generations. Improved cultivars, which are going to be more of the commercial line varieties that we see in large-scale agriculture, but also the wild relatives, both the wilder forms of the species or the crop species you're working with, and species that may be related and crossable to those wild species. All of these can form the basis for crop improvement and vital to basically our continued improvement and diversification of the food supply. If you were to go and look at the germplasm resources that may be out there, or if you were to go and collect germplasm, it can vary quite considerably off what we're used to eating in the supermarket. So this is an example of some of the variation you might find in grapes if you're collecting. For those of you who visited Geneva as the 406 class, you might have seen some of the diversity that's out there in the fruit collections in Geneva. This is an example of some of the apples that are in the collection. They can be anything from the size of a cherry to something that's pretty much the size of your head. Also, if you look at grapes, for instance, they're not going to be that typical bunch of grapes. Oftentimes wild stuff may be very, very different from what you would be used to seeing in commercial varietal types. But it's possible to take this material and find a lot of unique traits that can then be crossed into a commercial type, creating whole new market classes of crops, particularly in horticultural crops, where we've got so much variation in such things as color, size, shape, and flavor that we can really take advantage of. This is a new grape type that's being developed in Geneva, which is an elongated finger type of shape to the grape, which are quite nice to eat as well. They have quite a good flavor. But a lot of these variations can be very well received by consumers. So it goes beyond just the producer aspects of yield and disease resistance. There's a lot of consumer quality traits that can be selected out of germplasm also. I showed you the case of the red apple last Friday. Here it is again in a picture where you have a deep red flesh inside on the interior of the apple. So where does the US stand with respect to germplasm, germplasm collection, maintenance, preservation? Well, there was only a real serious effort started in agriculture coming up to about the time of the Civil War in 1861. This is about the time that state agricultural experiments were first proposed. And during the Civil War, Abraham Lincoln then put forward the foundation of the US Department of Agriculture. A lot of the state agricultural experiment stations were put maybe a little bit on hold because of financial issues associated with the Civil War. The initial site for New York's one was going to be in Willard, New York. It was eventually put into Geneva, New York, where it was opened in 1882. So we just had our 125th anniversary there this summer. But the USDA as a whole is not just responsible for agricultural research. It also administers a whole host of other issues, including conservation, food safety, food stamp programs, for example. In fact, the Agricultural Research Service budget is quite a small portion of the entire USDA budget. As part of the USDA, the importance of germplasm was realized, and in 1898, a plant introduction system was created for collection of germplasm. The first PI 000001 was a cabbage from Russia. And the term PI is short for plant introduction. That basically means that we take a plant from one place and introduce it to another place. So when we talk about materials that have been collected for the germplasm repositories, we oftentimes refer to them as plant introductions, or PIs, or accessions, because they're given basically an accession number. After the Second World War, this started to get taken a lot more seriously. There was a big effort in plant breeding at the time, and 3 regional plant introduction stations were opened in Ames, Iowa, Geneva, New York, and Pullman, Washington. In '58, the National Seed Storage Lab was opened down in Fort Collins in Colorado, and then the database for all of this material, or the GRIN base, was set up in 1978 to document all of the collection material and all the information that had been collected on those materials. More recently, a bigger effort's been focused on the clonal repositories in the 1980s, and in 1999, an ornamental plant germplasm center was opened in Ohio State, or in collaboration with Ohio State. Initially, a lot of the focus was really on the crops of major importance rather than the diversification of crops. So the plant germplasm system isn't just the USDA. It also comprises of a lot of other organizations or even small companies such as Seed Saver Exchange that go out and maintain a lot of this biodiversity. In fact, a lot of these kind of aesthetic traits you can often find more easily from sources such as Seed Savers than you can in the germplasm repositories. The USDA is known for its acronyms if you've ever been to one of their meetings. So they basically comes down to the United States Department of Agriculture, Agricultural Research Service, National Plant Germplasm Stations, Plant Genetic Resource Units. So you can actually get quite an abbreviation on some of their organizations. The germplasm repositories have expanded for political and geographical reasons. Griffin is now considered a regional plant introduction station, together with Geneva, Ames, and Pullman. And you can see that there's a number of smaller repositories that exist around the country, where, for instance, Palmer, Alaska, may keep some of the Arctic crop germplasm. I believe it also keeps the rhubarb collection. And Mayaguez, Puerto Rico, for instance, will hold some of the tropical germplasm. So some of this is based on a geographical aspect of it doesn't really make sense to try and maintain the papaya collection in Geneva, New York, knowing the weather patterns that we have there. But this gives an example about how diverse the holdings are throughout the 50 states and Puerto Rico. So when we're looking to utilize genetic resources, we have to consider how closely related they are to the commercial varieties we're looking to produce. Because as a breeder, if we can find that trait of interest in something that's closely related, that is always going to be the most preferable place to go. So ideally, we want to find our trait of interest in the primary gene pool, which means a gene that can be readily transferred through crossing in the case of seeds. So for instance, older varieties or land races that may have something of interest. If we're able to use the primary gene pool, then typically the number of deleterious genes and deleterious traits that we're bringing in is very much diminished. After that, we might want to go to the secondary gene pool, but as a breeder, we're going to start dealing with a number of difficulties here in that they're not readily crossable through sexual hybridization. We start ending up with problems including significant linkage drag and the fertility of F1 hybrids. This week I'm going to start talking about a number of these issues when I start talking about the use of interspecific hybridization in breeding, for example. The last place we really want to go, but sometimes it's the only place we can go, is the tertiary gene pool, which requires special techniques to be able to move that gene into the material of interest. So when we're looking to maintain these genetic resources in these germplasm stations, we're really looking at maintaining wild species, land races, improved cultivars, and more recently there's been more of an effort for breeding lines and genetic stocks. What you'll find oftentimes is a breeder may have a career of 30, 40 years that may have been built on another breeder of 30, 40 years, and when the breeding program is not replaced, all of that work in improving those crops can quite easily disappear, and has disappeared in many cases. So now if there's some very interesting traits that have come through in the breeding program, there's more of an effort to protect these. And also genetic stocks, which would include such things as near-isogenic lines, which would be very good materials for studying genetics of certain traits. So the germplasm centers within the US are quite expansive. They represent 185 families, 1,600 genera, over 10,000 species, and the total number of accessions held is about 460,000. This is a little out of date. It's closer to 480,000 now based on recent collections. But another issue with this number is that a number of these accessions may not be readily available to people requesting them. In fact, about 20 to 25% of these accessions would not be available to people requesting in the active collections because of lack of seed viability, for instance. The materials are stored basically in active or working collections. So most of the repositories increase the seed, store the seed, maintain the seed, and release the seed. These are known as working collections. Almost every repository has a working collection. Because some of the collections of species may be 30,000 accessions, this is very difficult for a researcher or breeder to work with. So in the last 20 years, there's been an effort to produce subsets of some of these crop species, particularly for those that have more than 1,000 accessions. So for instance, in the case of common bean or Phaseolus vulgaris, there's over 20,000 accessions available in the USDA collection, but there's a core collection of 406. That means if I want to look at a subset of common bean, I can order the core collection of 406 accessions that represents a lot of the geographical size, shape, color, maturity, a lot of the variation that exists in the total collection. The base collection then is a backup collection. This would be the Fort Collins collection. They are not released from these. They're basically here for long-term storage to make sure that that material is going to be have long-term viability. The base collection is based at Fort Collins, Colorado, formerly the National Seed Storage Laboratory, now called the National Center for Genetic resources preservation. If you ever have a chance to visit here, I would recommend you do. It's quite an impressive place to look at germplasm. They also do a lot of research on such things as the viability of germplasm and also the molecular study to look at for duplications, for example. But if we look at the National Plant Germplasm System, the activities, they're basically responsible for acquiring or getting hold of these materials, documenting them, passport data, where they're from, some of the interesting traits that may be present, for maintaining the seed or the clonal material, for doing some evaluation of these materials, and for distributing them. And in some cases, they have scientists based at the repositories who help with some genetic enhancement of the materials. As far as acquisition goes, this can be achieved through donation, exchange, exploration. If we look at the collection as a whole, 19% of all of the accessions held are native from the US, but over 80% of the materials held actually come from overseas. And they represent all three of the gene pools I talked about. When you acquire material, for instance, Phil Forsland's been acquiring material in the Central Asiatic region for apples. You can see in this case you've got some prior selection from a bear, and you can see the apple seed in the scat. Probably the bears are going to know their way around the woods better than we are, and they've done a lot of probably indirect selection over the years for apple evolution. This is also true of the Silk Road, with a lot of the horses and camels moving from the Far East down to the Near East along the Silk Road, where you have a lot of deposition of apple trees and apples that have been eaten along the way. So germplasm can be acquired in many different ways. But obviously we need to have a degree of documentation on where it's from and some basic evaluation and inventory data on that. This is where GRIN comes in. And if you have never accessed this site, I recommend that you do. I believe I have a web address for it. But it's where all of the information for the USDA collection is going to be documented. And now they have it in an improved version where you are able to go out and do general inquiries. So you could look up something like yellow tomato, and it might bring you back 10 accessions that have been documented as being that color, or heat-tolerant tomato. So if you're working on a particular project type, you might be able to do a quick search in the GRIN system and find germplasm that relates to your subject of interest for your studies. The storage of germplasm obviously depends on the type of germplasm. Seeds are typically stored with a low moisture content, down about 5 to 7%. For long-term storage, such as in Fort Collins in the base collection, they'll be stored down about -20, which is about the temperature of a deep freeze. But for the active collections, such as the one in Geneva, where the materials are going to be released to people throughout the course of the year, they'll more typically be stored at about 40 degrees Fahrenheit or about 5 degrees centigrade. In some cases, they will be cryogenically stored in liquid nitrogen at about -200 degrees centigrade. This is an approach in the long-term storage that's more commonly used for the clonal crops. And if you ever get a chance to visit Fort Collins, it's quite interesting to see their storage in liquid nitrogen. They have 3 enormous liquid nitrogen tanks that are probably 50 feet high. They use so much of it. But they're able to store clonally propagated crops such as apples just as a small piece of budwood in liquid nitrogen. This obviously saves a lot of time and money with respect to being able to maintain apple varieties, for example. A lot of study has gone into the rate at which these are cooled into liquid nitrogen, because that can have a big effect on the long-term viability of the material. In the case of Geneva, they're just held quite simply in seed packets at about 5 degrees centigrade, and then when the seed is requested, they just packet it out and send on. Yeah.
How often do they have to, will they have to like grow them out to reflect the samples, or will they just keep them there long?
It really depends on the crop type. Some seed has much longer term viability than other seed. If you look at crops like onion and garlic, it's got very, very short seed viability, maybe only 10 or 15 years. Other crops, especially the hard seed crops like alfalfa or cotton, could last for 100 years quite easily. So it really depends on the crop types, but basically their job is to continually be regenerating seed. So they periodically do seed germination tests, and they focus on the seed that is really reducing its germination. Because when a seed goes down, when seed viability goes down to about 80%, it quite quickly goes down 70, 60, 50 to 0. But it holds up at about 90 to 100% for normally quite a long time. But when it starts to drop off, it drops off really quickly. In the long-term storage, it'll be stored in the more vacuum pack type of approach, and again at about minus 20 degrees, not at 5 degrees. This increases the seed viability, but people aren't opening the packs to send out the seed all the time. In the case of clonal crops, I showed you an example of budding for a method of scion and rootstock propagation on Friday. So to store it, this should read 3.5 centimeters here. But a piece of budwood, meaning say a piece of apple wood with a bud on it, would be cut to about 3.5 centimeters. This can then be stored in liquid nitrogen for a long period of time. It would be stored in vats such as this in containers. This way, if somebody were then to ask for that variety of apple tree, because apples are clonally propagated, the budwood would be brought out of liquid nitrogen. We'd take a rootstock genotype, make a small incision, graft the budwood in, which would then start to germinate. We would remove the top part of the rootstock genotype, and then this way we can regenerate these apple trees from the budwood, which could be sent out to people requesting that particular variety. There's a lot of danger with holding living materials. In the case of Geneva, they try to hold 3 apple trees of every variety that they have, but we get problems such as fire blight and diseases will come in. The trees will die from abiotic or biotic stresses. So maintaining materials in this way is a very good approach to long-term storage, and it's much cheaper, of course, than keeping orchards going with all of these material. So as far as the maintenance, you have to do periodic germination tests, and accurate cataloging. Duplications can be a problem, but as I just mentioned a minute ago, some seed types have really poor long-term viability compared to others. The onion family is a very good example of this. So these require much more work to maintain. Also, on that front, open pollinated crops would require a lot more work to maintain. If we're working with a self-pollinating crop like soybean, then we just plant 100 of them and harvest them. But in the case of something like cabbage, which might be a biennial open pollinated crop, we have to set them up in cages with insect hives, etc., with about 100 plants for regeneration of the seed, and deal with the vernalization issues on it. So the cost of maintaining outcrossing and particularly biennial crops can be very high. The distribution is about 125,000 samples per year, most of it domestic, but about 25% of it is sent overseas, and this is a free service. Yeah.
What percentage of that is for other academic institutions or USDA institutions versus private individuals?
Most of it is probably within the public sector, because most germplasm enhancement is within the public sector. Private companies oftentimes work with very advanced materials, and they don't like to work with germplasm so much because it can be a number of years before that could be moved into a commercial variety. Companies are very interested about what their market share in a crop is going to be 5 years from now, rather than the long-term health of that crop variety or that industry. And a lot of this can be for screening of materials. About 3 years ago, I screened about 4,000 accessions of crucifers for a disease. So in that case, I would have been requesting about 3% of the total germplasm distributed that year. So this can be deceiving for the number of distributions, because you can have 125,000 samples, but you may only have 500 requesters for that material. It is distributed freely, but obviously with these active collections, they've got to package everything out and send it, so they don't like people to just say, can you send me everything I've got? Because then somebody's got to sit down and package out 3,000 different accessions. So they like you to give a reason for why you're requesting it, and also to give you some feedback on it, if that's possible. But the information for requesting can be seen at these websites, which are on the handouts at the back of the room. It's also possible, of course, to request germplasm from other areas. The European collection is becoming more consolidated. It was really based on collections within different countries. Now they're trying to bring it under one umbrella of the ECP genetic resources. You can access the site here, but a lot of these collections are still going to be based in the original countries. For instance, you might look up one particular crop type, and you might find that it's stored at Gardesleben in Germany, that it's stored somewhere in the Ukraine, that it's stored in a French collection. So they haven't really unified it that well, where you might actually find out there's 4 different locations that you could find a particular piece of germplasm you're looking for. But if it's not possible to find something from the US collection, this is a backup. I had this case recently, about a year ago, where I was looking for a navy bean variety Kentwood that was not present in the US collection, but was present in 2 of the European collections. And I was able to order it from there. Yeah.
African one as well?
They are on the international front. There are oftentimes individual collections in countries that have invested money in them politically. And if a crop type is important in that country, they might oftentimes have a collection of a particular crop species, but not an overall collection of many crop species. The main one in Africa is the Nigeria Center. A lot of materials have been collected from Africa, particularly the Ethiopian region. And a lot of those crops are then stored in particularly the CGIAR stations. This is the Consultative Group for International Agricultural Research. This group was set up in 1972 to try and preserve materials internationally for the benefit of everybody. It was focused primarily on the staple or carbohydrate type of crops. So the idea was that these stations would store and release germplasm to anybody in the world free of charge that was requesting it. And that they will not be able to then seek IP, intellectual property protection on this. The stations that were set up as part of this group basically involved about 8, I believe, institutes for edible crop plants and maybe 1 for forestry type of work. You may be familiar with some of these if you work with them. CIAT is in Colombia, CIMIT in Mexico. IITA, as I mentioned, is in Nigeria. That's the largest international African station. Oh, there's also one in Liberia, water for rice storage. Mainly the crops that are important worldwide on a calorie basis. So you can see the split up of the holding and then the types of crops that these different institutions actually hold and administer. This is the center in Belgium that holds the banana collection. Some of these, yeah.
Sorry, I was just asked, does the Millennium Seed Bank project hold germplasm, or do they...
Does the which project?
The Millennium Seed Bank project.
That's... I'm not sure what the Millennium Seed Project is. I know that they've had that one in Scandinavia recently where they're trying to put all of the germplasm into some subarctic zone that's free of any crop area. The British one presumably ties into the ECP type of approach. But the British have put a lot of effort in 2 collections. They have a number of collections that are quite expansive, particularly for horticultural crops. So that plays a critical part of the European collection. But they do tend to be specific with countries. For instance, if I were looking at brassicas, I'd find most of the Brussels sprouts were based in the Belgian collection. I'd find that a lot of the broccoli was actually in the HRI collection in the UK. And then a lot of the cabbage materials and other brassica materials might also be based at Wageningen Institute in the Netherlands. So sometimes they're more concentrated on with different crop species. But the European system, they're trying to unify it a little bit, but it's still a little bit disjointed because of the history and the nationality of countries in the collections, I think. Some of the problems that have really started to occur, though, in the last 10 or 15 years is people start to think that the germplasm that is collected from their countries is worth billions of dollars, which to some extent may resort back to the germplasm that can be used for pharmaceuticals, for instance. But a number of countries that have these centers of origin, Ethiopia is one in particular, have become concerned that the rest of the world is coming and taking their materials, but they're not getting any intellectual payback for this. You could argue that the food supply is, should be for the benefit of all internationally, and that breeding at the end of the day is. So trying to patent germplasm might be something like trying to patent other people from using your language, for example. So it does lack it does lack some intellectual grounding from the prophetic reasons. And in fact, the one reason that the USDA has a problem with granting intellectual property to germplasm is that that would essentially go against the US Constitution for granting basically intellectual property for something that doesn't really exist as intellectual property. So the US have not been a signatory on these recent international treaties on how to deal with plant germplasm. The first one was set up in the Convention on Biological Diversity in 1993, where there was a lot of talk about where countries utilizing germplasm would give some kind of payback to that country. And in fact, a lot of countries have actually signed on to that now, including most of the EU nations. I think 2 of the holdouts on that are the US and Japan, and most other countries have signed on these treaties. More recently, we have the International Treaty for Plant Genetic Resources, which the UN and the FAO are trying to administer, but it's really a situation that's still in flux, and they're still having meetings on over the last 5 years to try and decide whether there are any IPR issues related to this. But as part of this change in philosophy on who owns germplasm, when we have the plant explorations, which are overseen by the USDA, that is where a group will apply for a grant to go and collect germplasm from a particular country. In the last 10 years, we've started to see a lot of applications to collect materials in countries start to be denied. So it's now getting more difficult to collect germplasm, not just for political reasons, but also for just changes that have occurred in agriculture, where now so many people are actually just growing commercial varieties that are the same world over, rather than growing more traditional land races. So the ability to collect germplasm today is not what it was 50 years ago, despite the value that can exist in it. The utilization of these materials is obvious in food production, also in aesthetics, landscape, pharmaceuticals, more recently perhaps in energy. But the value of a particular trait can be astronomical. When Borlaug introduced dwarf wheat into India, the dwarfing gene allowed control of the height of wheat, which then enabled people to apply fertilizer. Older varieties of wheat wouldn't allow fertilizer because they would just fall over, because all that nitrogen would just basically make the whole plant structurally unsecure. This ended up tripling the wheat yields in India, possibly contributing tens or hundreds of billions to the world economy just based on one single gene. The ability to have that in collections is important. Somebody collected soybean germplasm from China in the late 40s, early 50s. It turned out that when cyst nematode issues became problematic in soybean, that this material had resistance to cyst nematode, two genes that has now been employed into soybean production worldwide in rotations, and it would be difficult to grow soybeans without them. The estimate for that, I think, is 100 million per year to the US economy, just from that soybean germplasm that existed in the collections. Obviously, there's a lot of concerns currently about the erosion of genetic diversity, how you should preserve materials, and who actually owns it. And these are ongoing and open for some quite heated debates at times. Morning. The lecture I'm going to give today may have a little bit of duplication, because I'm sure in this class you deal with the creation of hybrids to quite a large extent. But I want to cover some of the practical aspects of hybrid development, because that leads pretty well into the classes that I'm going to give on Friday of this week and Monday. I put this slide up last week just to talk about the gains in productivity that we've seen in, particularly in agronomic crops like corn, where the creation of hybrids, which was initiated around about the 1930s, we basically followed with the Green Revolution, and this combined with inputs like artificial fertilizer has really helped to boost yields in certain crop types. Other crop types maybe haven't increased their yields anywhere near as much as this, like ryegrass or potatoes. During that same period, a lot of the increase in them has come through perhaps inputs like fertilizer, but we haven't seen the effects of varieties. In many cases, it'll be claimed that breeding can contribute up to 50% of the gains that we've seen through the development of disease-resistant cultivars and hybrids. So really, why are we getting these gains from hybrids? And you might have already talked about hybrid vigor and heterosis in this. That basically, by taking two parents and combining them, we increase the heterozygosity in the F1 progeny. And through this, we can actually gain increases in vigor and yield. So the most common one covered, of course, is field corn. And as you're probably aware, field corn has the male flowers on the top of the plant and the female flowers in the middle of the plant. These are actually represented by corn ears here, but they should really be flowers. So the idea is that you have a male pollinator plant and a female seed plant. And when you harvest the F1 seed from this combination as a hybrid, it's going to have much more vigor than both of the parents. And this is, of course, the benefit, especially in crops where yield is very important. When we develop parents for hybrids, though, we tend to combine different attributes within each of the inbred parent. So, for instance, in the case of corn, if we look at the two inbred parents and then the hybrid, we won't necessarily be selecting the same traits in both the male and the female parent. For instance, we might be selecting a shorter, wider kernel in one of the parents and a more elongated kernel in the other parent, which will give then the best combination when you produce a hybrid. So this has really been successfully employed in corn. Corn is an outcrossing crop, and so it suffers from a lot of inbreeding depression. So when we develop parents for the hybrid cross, we tend to self-pollinate them. This increases the homozygosity of the inbreeding or the inbred parents, which leads to inbreeding depression. This is caused for a large part because a lot of recessive genes, when in the heterozygous state, they're really masked. So the more that you self-pollinate something, you uncover more and more potentially deleterious recessive genes. So you tend to build up a lot of negative genes in the inbreds, which results in the stunting of the plant growth and vigor. But over time, this has actually changed quite a lot. As inbreds have been selected for a number of decades, they've started to gain a lot of vigor as the deleterious recessive genes have been selected against in inbred parents. If you looked at the inbreds of corn hybrids, or the inbred parents of corn hybrids 30 years ago, they may have been this tall. Where if you look at the inbreds used to develop corn hybrids now, they may be closer to what the hybrids were 30, 40 years ago. So a lot of the work has really moved out a lot of these deleterious genes over a number of decades, and this has helped to, of course, further the yield and vigor qualities. how does this heterozygosity work to produce a more vigorous hybrid? This is a relatively complicated slide, which I have handouts at the back, and I'm sure you'll have a chance to look through it. But basically, if we just take the example of maybe 6 genes that might exist in each of 2 parents that you might be looking at to create a hybrid cross. Many of those genes are going to be homozygous, like AA, CC, DD, FF. Some of them are going to be dominant, some of them are going to be recessive. And some of the genes, like the B case here and the E case, will be heterozygous. So if you were to just take 2 parents randomly and cross them, the heterozygous genes in each parent wouldn't really match up in the F1 hybrid. So if we combined these 2 parents, you could see that the hybrids we produce are going to vary a lot at the B locus, and they're also going to vary a little bit at the D and the E locus. So this is going to result in segregation of those plants, or of those hybrid plants. So to reduce this so that we can gain not just more hybrid vigor, but also more uniformity, we need to self-pollinate these parents to create inbred lines or inbred parents. Every time we self-pollinate a plant, we reduce the heterozygosity by 50%, because a heterozygous gene such as BB, when this plant is self-pollinated, half of the possibilities are going to be homozygous recessive and homozygous dominant. So each time you self, the heterozygosity reduces by 50%. So in creating an inbred, if you were to self-pollinate 3-4 times, you'd be reducing the heterozygosity 50, 75, 87.5, 93.75, etc. So the more times you self-pollinate the plants, the more homozygosity you have at each locus. But the 2 parents can still be different at that locus with respect to the allele, whether it's a double D or the small d. So as we self this down, we may end up with this inbred line that has now become homozygous at all 6 of these genes. And the other inbred line has also become homozygous, but the allele might vary at any particular locus. So now when we cross these 2 inbreds to create an F1 hybrid for commercial production, we not only uncover a lot of heterozygosity caused by the differences between the 2 inbreds, but every F1 hybrid now is almost exactly the same genetically, even though it has a large number of heterozygous genes, which would not be the case here, where you have significant segregation if you have not selected inbreds for a number of generations. Yeah.
The plant on the left, I mean, it's gotten to the point where, say, like at the D locus, you have 2 dominant alleles there, and the one on the right, you have 2 recessive.
Yeah.
But it looks like, I mean, locus D is not. No, okay, with the B locus, because they're both heterozygous originally.
Oh, actually, this is a. This one's heterozygous. This one's homozygous.
Okay, so at the D or at the B locus.
Oh, at the B locus, this one was heterozygous, but we ended up selecting the inbred that has the small b type when we were developing inbred line 1. This was selected separately, and this was also heterozygous. But during the selection of this inbred, we actually selected the homozygous at the big B locus. So during the development and selection of these plants, we could have selected either a big B type or a little b type as we're self-pollinating. I'm just using this as a hypothetical example where in this case we selected a big B type, where in this case we maybe selected this plant and selected a little b. So they'll be different, but what happens when you combine them as an F1 hybrid, you now uncover heterozygosity at that locus, which tends to be fitter. But now you reduce all of the segregation, so you also have an extremely uniform plant. In fact, yeah.
I mean, that's general practice, though? You select for dominant homozygous on one side?
No, I think it's common practice that when you select inbred lines, you don't tend to select for exactly the same thing. So when you select for perhaps different traits, you are maybe selecting for different types of genes in one parent line to another one. For intellectual property purposes, companies like to have quite divergent parents. Because if you look at what we might find in the case of a hybrid, if we take an inbred that is maybe 90 centimeters and another one 85, we may get an F1 hybrid that has the vigor and is much taller. But if we go back to this slide, you'll see if you were to self-pollinate this F1 hybrid, then you'd create extreme variation by self-pollinating. So this enables companies to market an extremely uniform and vigorous variety. But if farmers try to collect seed from it in the field, the variation is going to be so great that it's not going to be practical to grow, and it's certainly not going to be practical to machine harvest. The benefit of hybrid seed is not just in hybrid vigor. It's not just in the ability to increase yield through that heterozygosity. As I showed you in the last two slides, the uniformity is extremely important. If you ever go out and look at some cornfields, what you're going to see is that every plant throughout the field is almost identical, maturity-wise, height-wise. This is extremely important for harvest efficiency, particularly with field crops where we're going to do mechanical harvesting. Because if we have variation in aspects like maturity or height, then it's going to be difficult to go in and harvest and maximize your harvest out of that. The intellectual property protection for companies is always also very important, because if you self-pollinate this F1 hybrid, you get so much segregation that you really cannot steal the materials from the F1. Another factor that can be quite important is the ability to introduce different traits into the two different inbred parents, particularly quantitative traits, which are difficult to work with. Because if you introduce different traits into the two parents, then you can combine those traits in the F1 hybrid. When I did my PhD, I worked in a tomato breeding program in Florida. And they'd had quite a successful F1 hybrid variety for spring production, fresh market production out of that program. One of the parents of that hybrid, 7324, was very tolerant to heat, which you need in a climate like Florida. It was also a very firm fruited type. The other parent, 7060, was a large fruited type and had higher taste and eating quality. So when these were combined into the hybrid solar set, solar set picked up some heat tolerance, some larger fruit size, some heat tolerance, and some flavor. So when it's difficult to select for a lot of quantitative traits, sometimes you can select for different traits, and then when you create the F1, you can combine those traits, which can really improve the variety at that point. It should also be noticed that hybrid seed production does not work in all crops. In fact, it's not used in many crops. Many of the large crops that are typically self-pollinating include wheat, soybean, rice, lettuce, common bean, although attempts to use hybrid seed production in many of these crops like rice and lettuce has been attempted. We are now starting to see hybrid rice and hybrid lettuce varieties come on the market. If a crop is naturally a self-pollinating type of crop, it's normally quite vigorous just through selection as a self-pollinator. Most hybrids that we see tend to be the open-pollinated species, such as corn or maize, which has airborne pollen, or the brassicas, such as cabbage, broccoli, which have insect transmission of pollen. It's not to say that self-pollinators cannot be used for hybrid production. Tomato is a classic example of that. Cultivated tomato is a self-pollinated crop. So you can actually get very vigorous plants just as open or self-pollinator. But many of the value of the seed is so high in tomato that many of the varieties that we buy are actually hybrid because it's cost effective enough to produce the hybrid seed in tomato. Yep.
How do you produce the hybrid seed in tomatoes? Do you have a male selective?
I'm going to cover tomatoes quite a lot during the lecture, and I'll come on to some of those aspects. With the value of the seed being so high, if you want to, you can produce it with just hand pollinations, because one pollination could produce maybe 100 or 200 seed in a fruit, for example, and you just have to have a cost-effective enough labor force. But there are other methods, such as male sterility, that are and can be used in tomatoes, and I'm going to come back to that because I cover that quite a lot in the lecture. So when we're looking at the generation of hybrid seed, the three most common methods of producing these hybrids, which is important for commercial seed production, is the mechanical approach, such as detasseling, hand pollination, or even chemical or hormone type of approaches. Self-incompatibility, which occurs naturally in outcrossing species to prevent self-pollination and to encourage the outcrossing, or male sterility. These are the three most common approaches used for hybrid seed production. When you're looking at any cross, whether it's for developing populations as a breeder or whether you're looking at hybrid seed production, it's important to know some real simple botanical facts, particularly to know the type of flower that is present in the crop species you work with. This here is what we would describe as a perfect flower, in that it is hermaphrodite, having male and female parts, the carpel and the stamen being the anther and filament, and the carpel being the pistil and the ovary, and then it has the petals and the sepals. So a perfect flower has all four of those components: carpels, stamens, petals, sepals. This would be the case in something like a tomato. In the case of corn, it would be imperfect flowers. So if we were to look at the tomato flower, why is cultivated tomato a self-pollinator? Well, if you look at the growth of the pistil, the anther sac, which is this large yellow piece in the center of the flower will produce pollen at the top of the anther sac. The female carpel produces the pistil, which, before the flower opens, the pistil grows through the anther sac and pollinates itself before the flower opens. So thus it's basically a self-pollinator. So the pollen is already producing pollen tubes which are going down to fertilize the ovules before the petals open.
Why does the tomato plant go through all the trouble to make petals?
Naturally, tomato wild relatives are open pollinated. As the wider related species, they tend to be outcrossing. It's just cultivated tomato, which has now been named, I think, Solanum lycopersicum. It's the only of the Solanum tomato species that is actually a self-pollinator. All of the related species are actually outcrosses. So it's probably been selected for this over time through domestication. And basically, as we can see here, the pollen tube fertilizes the ovules. The ovary ripens into the fruit, and the ovules then are basically the embryos that form the seeds in that fruit. In the case of something like squash or cucurbits, they typically have male and female flowers on the plant. They're great for crossing because the flowers are huge, about the size of your fist in many cases. So in this case, it might be possible, for instance, it's an insect-pollinated plant, to bag the female to prevent it from becoming insect-pollinated, and then be able to take the anthers and the pollen from the male of interest and mechanically pollinate, and then rebag. In the case of cucurbits, it's also possible to use some chemicals and hormones to induce plants that will be entirely male or entirely female flowers on the plant. This would be considered monoecious as opposed to dioecious plants in where one plant is male and another plant is female. It's still monoecious in the fact it has male and female flowers on the same plant, but they are imperfect flowers. I want to quickly show you some crosses of vegetables and horticultural crops, because the basis of any population development and any breeding you do is in the cross, and this, of course, can vary quite considerably from plant to plant. So what we can see here is a broccoli plant. The broccoli has now gone on to form the racemes with flowers. And if we want to do a cross here, this is actually going to be a self-pollination of a self-incompatible plant. The immature bud is being opened up. Self-incompatibility develops as the flower matures. So in the last two days prior to opening, the self-incompatibility starts to increase because it's chemical-based. So by the time a flower is mature and open, if you pollinate it with a flower from the same plant, it'll be self-incompatible and it won't go on to form any seed. In this case, an immature bud was opened and it was pollinated with pollen from the same plant, thus overcoming self-incompatibility. This is the kind of approach that can allow us to develop inbred lines in outcrossing species. Broccoli is an outcrosser, insect pollinated. It will not pollinate itself, typically because of incompatibility. But by using this approach known as bud pollination, we are able to overcome self-incompatibility for breeding purposes. This now is looking at making a tomato cross. This can be done with electric toothbrushes or other means, but the anthers, sacs, are being pulled off the immature flower before the pistil pollinates itself. This can oftentimes be left for a day before pollination, and then we'll take pollen that's been collected from another parent, and this way we can make a cross or pollinate a self-compatible plant. So as I showed you with the tomato, when we have the anther sacs, the pistil grows through and pollinates itself. In this case, we take an immature flower before the pistil has done this. We remove the anther sacs to prevent it from pollinating itself. And then we take pollen from another plant, put it on the pistil, and that way we're able to make a tomato cross.
How successful are these processes?
As long as people are picking the right sized flower, they're very successful. Ideally, you do it in a controlled environment, not in the field. You probably have a little bit lower success in the field. Occasionally, you'll get a self-pollination, but there's tolerance for about a 2 to 3% self-pollinations within a hybrid lot. If you're moving towards about 10% self-pollinated, then that becomes typically unacceptable. So the occasional self-pollination is okay. And we make quite a lot of crosses with tomatoes for color and shape, and occasionally we'll increase an F1 and plant it out as an F2, and it turns out that it was a self-pollination of the female plant. But maybe one time out of 50. Depends how good the people are working with it, and of course if you work with this a lot, you're going to be much better at it. This is Joe Shale, who used to work with Dick Robinson, vegetable breeder with lettuce, and he's actually talking about the process of breeding lettuce here, about making a lettuce cross. And the flowers basically develop first thing in the morning, so you basically have to do the lettuce cross before about 8:30, 9 o'clock in the morning, or it will have self-pollinated itself. But he actually speaks on this one, so I'll let him describe it.
But I'm going to cut this one for demonstration, and I cut it just like so. Just above the green. Yeah, right above the petals. And then we wash it.
That's removing the anther sacs.
I try to wash it first. And I wash it very aggressively. If I'm going to hurt the flower, some people are a lot more aggressive than I do, in fact. And that removes pollen that's there, and any pollen that's about to shed. In fact, I often wash them several times, and then so the time between cutting and washing and pollinating can be half an hour. The warmer it is, the faster. Today's fast there. Today's fast. I anticipated that. But these flowers are in a good stage of pollen. We'll just start, you can even see the little tiny feet on there. I don't think your fixed line is quite shedding pollen yet. We'll take one of those flowers, look at it in the microscope. I don't think fixed line is quite shedding pollen yet. We'll just grab one of those in there. The same is true for these columns on this plant.
Matt's never made a lettuce cross before, so that's what it
But let's just take a better look at this. She brought an extra pair of scissors with us. They could just cut the extra cuts up so much easier. Yeah, here they are right in the center. I don't see. There you go. When they have my spare scissors with me, I didn't have my pollination here. Yes, in fact, here they are right there.
So he's finding the mature anther sacs from the other parent plant.
There's right where we want to be. And so let's just assume that everything is ready. We hold it as right
That's another example of making a cross with a self-pollinating vegetable such as lettuce, mechanically.
Trying to be gentle. But somebody told me once, should I slam it on there? I go, well, slam may not be the word that you want to use. So place it. Right. When you do that, then you have it right where you want it.
So that would be a lettuce cross. A bean cross, we can actually separate the male parts from the female part, and we can actually pull off the anthers and then just twist them into the female pistil for the cross. I think this is relatively quick, so I'll put this one in also. So you take an immature flower before it self-pollinated itself. It's opened up, and the male anthers are pulled away from the pistil. And then we can take the anthers, the mature anthers from a mature flower on a different plant, and we can then hook that into the pistil to pollinate it. Every plant is different. Some plants are much easier to cross than others. I have a reference at the end of the lecture of a book, Hybridization of Crop Plants by Fair, and that goes through almost every cultivated crop plant, and it goes into detail about issues like flower structure, methods of pollination, and how to do crosses. So if you are going to be crossing with your plant, it's good to look up the chapter that will relate to. In the commercial setting, you might well have seen this kind of setup in fields around New York. Oftentimes we'll have 6 lines planted of female corn seed parents or seed inbreds, and then we'll have 2 rows of the male pollinator inbred. These will be perhaps planted at different times if there's a maturity issue, ideally at the same time. But the female plants that are going to be used for seed harvesting will go through with a machine, and they will be detasseled before the pollen is mature. So the male flowers at the top of the corn plants will be removed mechanically so that they're unable to self-pollinate each other. And then the 2 rows of the male pollinators, as you saw in the first slide, these will then produce the pollen that will pollinate the kernels, and then these may be removed from the field and the hybrid kernels will be harvested. This would be hybrid seed production of corn in the mechanical approach, not just growing. Oftentimes the males are cut out so that you don't get the seed mixtures. So that is an example of how commercially crops can be, or hybrid production can be approached from a mechanical side. The reason we see it quite a lot in New York State is there's a lot of sweet corn grown here, and a lot of the sweet corn hybrids are produced through mechanical detasseling, not through male sterility, which is more common in field corn. But the 2 most effective approaches to hybrid seed production, the first one is self-incompatibility. Open pollinated crop species are self-incompatible, meaning that their pollen will not pollinate or will not fertilize the ovules of that plant. This aspect is used in hybrid production, but it can be based on a reaction of the pollen grain at the pistil surface or a reaction of the stigma at the top of the pistil. So the incompatibility can be from the male side or from the female side. When the pollen lands on the stigma surface, there's a cell interaction, there's proteins. Obviously, I don't have time to go into self-incompatibility in depth, but it involves 2 or 3 major enzymes, S-locus kinase, S-locus glycoprotein, and the interactions of these actually cause self-incompatibility when the pollen meets the stigma surface. I said that this can be determined by the pollen grain or the male gamete, or it can be determined by the female flower. If it's determined by the pollen is a gamete, it's a haploid. So this system is known as the gametophytic self-incompatibility. You can have up to 2 types of self-incompatibility genes expressed. A plant may be homozygous for S1, for example. It may be homozygous for S2, or it may be heterozygous for different self-incompatibility responses. In the case of brassica vegetables, they have over 50 S alleles. So you might be working, one parent may have S47, S49, the pollen parent may be S18, S20. They could be heterozygous or they could be homozygous. In the case of the gametophytic system, if the female flower was S1, S2, it would prevent S1 and S2 pollen grains from fertilizing it. So its own pollen grains would not be able to fertilize the flower. If it was S1, S3 at the flower, the S1 pollen grain would recognize it as an incompatible reaction, and it would abort its pollen tube growth. The S2 pollen grain would not, and it would go on to fertilize the plant. So the incompatibility here is determined by the pollen grain, and if the pollen grain is different from the diploid female flower type, then it will go on to pollinate. So if the female flower was S3, S4, both S1 and S2 pollen grains would both form pollen tubes. If the self-incompatibility is determined by the female pistil or by the stigma surface, it's a little bit different because now it's controlled by the female. So in this case, if the female was S1, S2, it would prevent S1 and S2 pollen grains from germinating. But if the female was S1, S3, it would prevent now S2 pollen grains from germinating, even though it doesn't share a gene with the female flower, because the S1 pollen grain has initiated the self-incompatibility of the female flower, and now the female flower prevents the germination of any pollen grains on the stigma surface.
I thought it had to do with the protein coding on the pollen grain, which carried both alleles.
It does have to do with the proteins at the cell surfaces, but the proteins expressed by the female flower at the stigma surface, you can have 2 that are expressed in the female because it's diploid. In the pollen grain, you only have 1 because it's haploid.
I thought you had
Well, it might well be, but as pollen types, the real aspect is that the reaction, is it in the pollen grain or in the stigma surface? All diagrammatic forms I've seen have always given the pollen one type of genetic base as a haploid. And typically with an inbred, of course, you're going to have homozygosity at that locus anyway. Yep.
So then, would that flower never be able to be pollinated then, from that point on?
In the presence of the pollen grains that are initiating the self-incompatibility. Because if the female flower surface recognizes that pollen as being the same genetic type or the same incompatibility type, it initiates an incompatibility response where it prevents germination of pollen at that surface. So it would probably, so that would be a dead end, most likely, because it's in the presence of a like S allele. And again, this is just a more colored example of the female type, where in the gametophytic system with the pollen, if the pollen is not the same type as the flower, it's able to form pollen tubes. If it is the same type, it is not. The use of self-incompatibility is widespread. It is not perfect for a number of reasons. One of those reasons is that even in highly self-incompatible plants, you do get a very small amount of self-pollination, where the pollen grain grows and fertilizes irrespective of self-incompatibility. So if you have a self-incompatible plant, you may have a couple of seeds form on that plant, where if it was self-compatible, you maybe have 1,000 or 2,000 seeds form on the plant. So using self-incompatibility, the strength of that system can vary depending on the parent or the genotype you're using. And you can end up with a small number of selves. The risk of this for hybrid seed production, of course, is that if people want to steal the inbred parents, if they were developed in a self-incompatible system, you can just go out to a grower's field and pick the small plants in the field, and then the likelihood that would be one of the inbred parents. So it's a way to basically steal the intellectual property of the commercial seed company if you're able to select the smaller inbred fields out of a commercial field. This is why they prefer not to do two-way crossing with incompatibles. They prefer to have a male and a female plant, because if you do two-way, then people can potentially steal both of the parents from the field that are used to create the hybrid. Male sterility tends to be a much more effective system and is much more preferable for hybrid production. Male sterility is based on basically dysfunctional male parts of the flower. The systems typically used are cytoplasmic and nuclear male sterility. In some cases, we can also select and find functional male sterility, where, for instance, the anthosac never dehisces, so it's never able to produce viable pollen to pollinate itself. In the case of cytoplasmic male sterility, it is controlled typically through cytoplasmic genes, oftentimes in the mitochondria. I think in the case of the T cytoplasm case in corn, with southern leaf blight, it was the URF13 gene in the mitochondria used, for example. But we can also have male sterility that is governed by nuclear genes. But it is these two approaches that can be employed in the production of commercial hybrid seed. If we were to look at a flower of a male sterile plant, oftentimes the flower will be smaller. It may have very small, sometimes have no anthers at all. And if you touch a flower, oftentimes you can get pollen on your fingers, a mature flower. In the case of a male sterile, you can rub it and you won't see any pollen. But a lot of the time, there's a lot of other flower attributes are diminished in male sterile types, which can cause problems in seed production. If we were to look at a corn plant, we can see the male flowers of a male fertile plant, and then the male sterile type. So morphologically, male steriles can be differentiated from male fertile types when selecting parents for a cross. In the case of cytoplasmic male sterility, as I mentioned, it's usually mitochondria-based and oftentimes has effects beyond the male sterility itself. Pleiotropy is one gene affecting the expression of other genes in the plant. This can also be created in the lab through transformation approaches. This has been shown in tobacco using a fatty acid interference gene. In the case of cytoplasmic male sterility, we basically develop the seed parent or the female parent, which is cytoplasmic male sterile. So we have to keep recurrently crossing and pollinating onto that seed parent to produce an inbred that has male sterility. We then develop a pollinator, which is going to be used to develop a hybrid. This is fine in the case of a crop like cabbage, because cabbage is a vegetative crop. In the case of cabbage, if we create this hybrid will also be male sterile. In the case of a crop that produces, for instance, a fruit, then we also need to have a restorer gene, which is usually nuclear, and this restores the sterility in the hybrid so it can self-pollinate itself or be pollinated to go on to produce the hybrid type. This is illustrated in this diagram where we have the sterile cytoplasm, but we have a restorer gene in a normal cytoplasm, and when we create the hybrid, the dominant restorer gene restores the fertilization even though we have the cytoplasmic male sterile cytoplasm. With nuclear male sterility, we're just dealing with nuclear genes. So there's a little bit of difference in how we might set up the parents for crossing. This has also been achieved through transformation, as you can see in the case of canola. If, for instance, we have a single gene recessive for nuclear male sterility, we may recurrently cross, for instance, onto the recessive male sterile type and then keep selecting the male sterile plants. We need a heterozygous, so we need a heterozygous parent in order to ensure that we get a 50% segregation of the male steriles. and then the male sterility here can just be restored by producing a hybrid that's heterozygous at that locus. This oftentimes involves the use of isozyme or molecular markers, for example. A nuclear male sterility option is now being used in canola. It's been examined a lot in tomato. In corn, it's mostly cytoplasmic male sterility. In brassicas, brassica vegetables, it's mostly cytoplasmic male sterility. There are a number of problems that occur with flowers, such as the lack of development of the nectaries, or petaloidy. This causes a lot of problems in seed production, where we're using insects to increase the seed, because the bees do not like to visit the flowers of male sterile plants. This results in really low seed set and really inefficient seed production. We can also end up with smaller petals or smaller flowers, some temperature sensitivity, and in some cases, we may not have a restorer gene that will work with the male sterile system. So these mating systems are really vital, and they can basically be used to get efficient production of hybrid seeds, which is much more efficient than hand pollinations or even mechanical for that matter. I realize I'm probably covering at least 2 lectures within one at this point, but it is an area that I believe Vern is also going to cover. So it's not the only exposure that you'll have to it, but I wanted to cover it quickly and briefly because I believe it really pulls in some extent into the lectures that I'm going to cover on Friday and Monday on interspecific crossing and polyploids in crop production.